And I know for many of you panelists, 2020 really represented a culmination of 2 decades or more of work, really seeing these first in human trials become a reality. So tonight we're excited to get a glimpse into your labs and the exciting research that you all have going on. I'm Liz Gelazzo. I'm the clinical director for the Angelman Syndrome Foundation. I'm a pediatrician, and I'm also a parent to a 6-year-old, Evelyn, with Angelman syndrome, and I'll be your moderator tonight. I'll be guiding the questions and the discussion and reminding all of these folks to speak English and not medicalese as best we can. We'll be focusing on the work that you're all doing rather than the ongoing clinical trials. As exciting as they are just that. They are ongoing clinical trials. So we will not be focusing on that tonight. So we're going to jump right in. The way this is going to work is I'm going to start by going around and asking folks to introduce yourselves. There are, I imagine, new families on the webinar tonight in their home where you may not be a regular household name. So I'd love for you to introduce yourself, tell us where you are, what your lab focuses on, and what you're excited about right now that you're working on. So I'm going to go clockwise around my screen. And so I see Steve Gray up in my top corner. So Steve Gray is an associate professor at UT Southwestern, and I'll turn it over to you to introduce yourself.
Yeah, so if people don't know, UT Southwestern's in Dallas, Texas. I moved here about 3 years ago from UNC Chapel Hill. So I know Ben and Mark really well, and Stormy, and really it's like a family reunion here. But my background and expertise is really around gene therapy, around using a viral vector called AAV to deliver genes to the CNS. And I've collaborated with Ben on some projects looking at trying to develop a gene therapy approach for Angelman syndrome. And I'm also involved with kind of drug manufacture of these viral vectors. I direct a core facility at my university, making these vectors to go into animals or patients.
So one thing that I'm going to do as we go is you guys say buzzwords. I may stop you and ask you to explain buzzwords that you're saying, because these are words that the community is now getting kind of a crash course in drug development and research, right? So when you talk about a core facility, what does that mean?
Yeah, no, it just means that it's like a sort of nonprofit facility within the university that has a mission to perform some kind of service to the university at large and sometimes to other clients outside of the university. So in this case, we actually produce these viruses, these therapeutic viruses at the university, and we use them for research studies or manufacture them actually for use in like early human clinical trials.
Great. So like phase 1-2 clinical trials before. Yep. Okay, great.
Yep.
Perfect. Thanks. All right. Eric Levine is a professor at UCONN. Tell us about yourself and how you ended up here, what your lab does, and what you're excited about.
Hi, so I'm Eric Levine. I'm in the neuroscience department at the University of Connecticut Medical School. And my lab is really sort of basic science, basic physiology lab. And we really got interested in trying to understand how UBE3A works in neurons to cause the functional changes that it does in neurons. And this really grew out of a very strong and ongoing collaboration with Dr. Stormy Chamberlain, who you'll all hear about, who generates these stem cell lines. They're called iPSC-induced pluripotent stem cell lines from individuals with Angelman syndrome. So it allows us to study in my lab at a cellular level the functional changes in neurons, because even though we know that UBE3A is the gene that loss of function of which is what causes Angelman syndrome, we really don't understand how it works in neurons to produce those changes. And so we think that by understanding the pathways and the molecules that it targets will really open up a much larger window of potential therapeutic targets that could be exploited for drug development. And one of the projects we're particularly excited about is actually using these cell lines to try to understand the roles of some non-UBE3A genes in Angelman syndrome. We know that Angelman syndrome is usually caused by a fairly large deletion that not only knocks out UBE3A, but knocks out several other genes who are going to get a partial reduction in those genes. And the interesting thing is that there are forms of Angelman syndrome that are caused just by the loss of UBE3A without those extra genes. And those individuals usually have a milder form of Angelman syndrome, especially with regards to seizures and maybe anxiety and motor symptoms. So we hope to be able to use cell lines that we can, in collaboration with Stormy, from individuals that have these different genetic forms of Angelman syndrome to try to uncover roles of some of these other genes. Again, would increase new novel therapeutic interventions that could be very useful for certain aspects of Angelman syndrome. And the possibility also that maybe these targets would have a longer window of potential effectiveness. We know that restoring UBE3A is only effective if it's done very early in development. And so maybe these would offer some other opportunities for therapeutic development.
Awesome. All right, break this down for us here. It sounds like you're talking about some sci-fi stuff. You're talking about making iPSC lines. So what does that process look like from taking a patient sample to making a immortalized cell line?
Well, I should probably let Stormy address that when she talks, because she's really the one who has pioneered that approach, and I'm just taking advantage of it to do our experiments, but I'll let her give a little more information about that.
Perfect and totally fair. Feel free to punt questions to one another. All right, and Stormy is next up in my rotation here. So Stormy Chamberlain is an associate professor also at the University of Connecticut. So I'll let you introduce yourself.
Yeah, like Eric, I'm at the University of Connecticut School of Medicine. I also chair the Scientific Advisory Committee for the Angelman Syndrome Foundation. I'm in the Genetics and Genome Sciences department. In my lab for a long time, like before I had a lab, I studied the gene regulation on chromosome 15. And really as a graduate student, I started asking the question of how does paternal UBE3A get shut off? And I must be a really slow scientist because it took, like 20 years to kind of understand how this actually works. And so my lab uses iPS cells, induced pluripotent stem cells, like Eric mentioned. And these are cells where we can take a sample, usually it's a skin punch biopsy, or more commonly now a small blood draw from a patient. And we add virus to them. In this case, it's Sendai virus. It doesn't integrate into the genome, not a coronavirus either. And what it does is it really hijacks the program of these cells and turns them into a stem cell. And the reason we do this is because we can turn stem cells into neurons. And then I can hand those neurons off to Eric, and he can do his great work with them and understand what goes wrong. But my lab, we try to make different deletions. We use CRISPR engineering to delete things and move things around to try to understand how does dad's copy of UBE3A get shut off? Because by understanding how it gets shut off, we can figure out ways to turn it back on. And one of, I think, my greatest joys is the tools that I've made, I can give to others, and they can use them to do really great things. And I don't necessarily even have to be involved. And so it's pretty tremendous. And I think one of the other really cool things that comes from our work is we can, every now and then there's a rare patient that has a really unique genetic makeup. And those patients sometimes tell us so much about the disorder and about how gene regulation occurs. And, we've been able to a couple of times take advantage of finding these patients through social media or other connections and using them to answer important questions about Angelman syndrome and some of the other disorders on chromosome 15.
Cool. Can you give us an example of a patient that might have been unique in some way that led to something that you learned that was different?
Yeah. So one of the keys to helping us understand how the human antisense transcript, so dad's copy of UBE3A is shut off by a long non-coding RNA. And one of the keys that told us how that's regulated in humans came from a patient with Prader-Willi syndrome due to an atypical deletion. So they had, most of the time, patients with Prader-Willi syndrome have the exact same deletion that kids with Angelman syndrome do. It's just on the other copy of the chromosome. And this one was a little bit different. It was smaller, and it took out a boundary element that made that patient's cell imprint UBE3A even in non-neurons. So that whole process only happens in neurons in humans. In this patient, they did it in blood cells. And so we were able to take that patient and really try to understand how that works. And that was one example of a rare patient that helped us.
Very cool. Very cool. All right, thank you. All right, going around my screen here, Ben Philpot, who's a professor at UNC Chapel Hill.
Yeah, hi, I'm Ben Philpot. As Liz said, I'm at the University of North Carolina in Chapel Hill. I've been working on Angelman syndrome for the better part of 15 years. My lab's research is really kind of divided up in 3 different areas. One is just to gain a basic understanding of what the role of the UB3A gene is for the development of the brain and how it impacts the development of synapses, which are the connections between brain cells, and also circuit development in the brain. So it's kind of a basic research understanding. And then there are 2 preclinical translational efforts that I'm really trying to bring forward. So one is a gene therapy effort that I've been working on with Steve Gray for a while, and recently partnered up with AskBio to try to move a gene therapy forward for Angelman syndrome. And the other is trying to use a small molecule approach to try to unsilence that dormant copy of UB3A. And this is something that I'm working on in collaboration with Deerfield/Pinnacle Hill, as well as with Stormy Chamberlain. So, we're really focused on, all 3 of these efforts and trying to push forward in all 3 areas at the same time.
Awesome. All right, break that down for us a little bit further. When you say small molecule, what are you referring to?
Yeah, a small molecule is really just a drug. So a drug that can be taken orally or delivered intravenously, any type of route of delivery. But a drug that could get into the brain and do what we needed to do, which was to, turn on that dormant copy of UB3A.
Awesome. Awesome. Great. All right. Next up, I have Jason Yee, who's an assistant professor at WashU.
Hello, everyone. I'm Jason Yee. So I have kind of a diverse background. The first was studying brain development in the embryo, and the second is in protein biochemistry. And so my lab kind of combines these 2 interests and trying to understand how proteins contribute to regulate nervous system development. So I began studying UB3A, I guess about 10 years ago during my postdoctoral fellowship with Mark Zylka at Chapel Hill. I started my lab at WashU about 4 years ago. And for the past several years, and this is work that's been funded by the Angelman Syndrome Foundation, we've been studying missense mutations in UB3A, which are mutations that change usually 1 amino acid in the enzyme. The importance of this work is that it's usually hard to determine if a missense mutation causes a disorder because sequence variations happen all the time between perfectly healthy individuals, but if that mutation happens in the right place, it can be deleterious. So there are really 2 goals for our work. The first is to identify the missense mutations that are likely disease-causing in UB3A. And the second, we think that if we can identify those disease-causing mutations, it will tell us something about that portion of UB3A that is important for its function. And we think that is a good gateway to help us understand what it's doing in the cell.
It's really fabulous. There's so many more patients who are being diagnosed with UB3A mutations as we're sequencing more patients. So understanding that mechanism as well is really critically important. All right, and last but not least, Mark Zylka, down here in the bottom of my screen, a professor at UNC. I'll let you introduce yourself. You're on mute. Yes.
Got it. Good. Yeah, I'm used to being last. With the last name Zylka, I was always
Sorry.
It was easy to figure out where to go in line in elementary school when you had to line up alphabetically. I just waited till the line formed and stood at the end. So anyhow, I'm at UNC. I'm the director of the Neuroscience Center. And as far as what my lab does, I really got interested in Angelman syndrome as a result of a collaboration with Ben Philpot. And I think, at some level, it's good to put a little history behind what we're all talking about today. Because I think a lot of the parents out there are probably very excited about the ASOs and the trials. And we know that these ASOs work by unsilencing the paternal allele, but it wasn't that long ago that we had no clue that a drug could even be developed to do that, to turn on the dad's copy of the gene. And years ago, I worked with Ben and another colleague, Brian Roth, and we set out to do this drug screen. And this was a screen that was funded by the Angelman Syndrome Foundation. And we basically looked at over 2,000 drugs and found a class of drugs that could indeed turn on the dad's copy of the gene. So this was really an important discovery that was funded by ASF. And had that discovery not been made, it's questionable as to whether or not ASOs would then be developed. Because when you look back at the initial ASO paper from the Baudet lab, the positive control he used was the drug that we identified in our study. So I think part of what I hope you all get out of this is not only what we're doing in the lab, but where a lot of these seminal discoveries and where this ASO ultimately came from. And so one of the things we're doing now to really build on the work that we did with these small molecules as well as with the ASO work is to try and come up with a way of essentially building on the ASOs. The issue with the ASOs is that they're going to need to be injected repeatedly every 4 months. And we want to try and find a way of essentially getting the benefits of an ASO, but doing it with a single shot. And so Steve Gray, for example, knows a lot about gene therapies. People talk about these gene therapies as being once and done cures. You inject an AAV gene therapy and you never have to do it again. And so we're taking advantage of this molecule called CRISPR-Cas. You probably heard a lot about it in the news. The Nobel Prize was awarded just this past year for the discovery of CRISPR-Cas and genome editing. And we found a way to use that technology to unsilence the paternal allele. And the nice thing about this is you can inject it one time and it lasts, as far as we can tell, a lifetime. And so this is something that we're very excited about and taking what we've been doing in mouse and turning it into something that works in humans.
Fabulous. So let's go back to a question about something that you just mentioned. So we get this question a lot about those people go back and they look at those initial studies that identified topotecan as a drug that could unsilence the paternal allele. And we get the question often, why can't we just use topotecan then? So maybe I'll direct this back to Ben.
Yeah, I think it was wonderful to demonstrate that topotecan could unsilence that dormant allele of UBT1A. But if everyone has ever had the unpleasant experience of watching someone go through chemotherapy using a chemotherapeutic like topotecan, because that's what it is, we know that they have unpleasant side effects. So there's a loss of white blood cell count, there's nausea. There are a lot of side effects of that. So that's one of the issues. And the other issue is it turns out that particular drug doesn't get into the brain particularly well. And also that once it gets in the brain, there are pumps in the brain that pump things out of the brain to keep it safe. And one of the things that it pumps out is topotecan. So it gets actively pumped back out of the brain. So that means that it doesn't get everywhere in the brain like we need it to. So an important goal is to search for drugs that have better penetration into the brain and that don't have the side effects of topotecan. So that's a very important goal for the field and something we're working on.
Yeah. Are there strategies to mitigate this issue of drugs being pumped back out of the CNS space?
Yeah, this is really a space where medicinal chemists can work on to try to optimize strategies to make it so that the drugs aren't recognized by the transporters that take things out of the brain that you want to keep there. So it's why really a field like research where you're trying to develop a therapeutic, you have to bring in a lot of expertise from a lot of different players. So this is something where my lab doesn't have expertise in. So we collaborate with a medicinal chemist by the name of Jeff Obey, and of course take advantages of the pharmaceutical industry and their might in this area to try to develop better drugs, basically.
Gotcha. And we're going to get back to this issue of the team approach that's necessary, but I want to take a step back and hit on something. This is really a question for all, but I'm going to start with Jason because you mentioned this. How has ASF, pilot funding or those initial pilot grants you've received from the ASF allowed you to either, get larger grants or attract funding of other funding bodies like the NIH or other private foundations?
Yeah, so funding's tough. So as a new investigator, I think it was really critical. So getting that grant from the ASF allowed us to do enough of the research to carry it forward to generate enough of the preliminary data in order to apply for larger awards. So I think I got the award almost 2 years ago now, about a year and a half, something like that. And a lot of that work that I had done went into my R1 application for the NIH, which was funded a few months ago. So that, without that, it would have been really hard to sort of do the amount of research that we needed to get those bigger grants. So that was critical.
Awesome. Congratulations.
Thank you.
For those who are not familiar with this NIH structure of funding, what does an R01 mean? What's an R award?
So that's sort of the standard research grant from the NIH. So the R1 is usually the standard which most academic labs sort of run their programs. It lasts for 5 years, and that's, it sort of gives us the resources to do the research for that period of time.
Yeah. And maybe for Stormy, can you help us understand like what the scale of that funding might look like? The pilot funding that you might get from a private foundation versus what you're then going after from a larger funding source.
So at ASF, our grants are, up to $200,000 to be used over 2 years. And this is really it's really sufficient funds for most of one person and supplies to do work that might take 2 years. Usually it takes a little longer. I think ASF gets a good deal for its $200,000. And then when we turn around and then ask for an R01 from the NIH, like Jason said, that's 5 years, and you might be able to get 2 or 3 people paid for to work on a project over that 5 years. And so the amounts of those, they're typically somewhere between $250,000 a year to $500,000 a year in direct costs. So, and then the NIH pays the universities a little more on top of it to pay for lights and snow plowing in the driveways and things like that. So the total amount of those, and that's important for us in a university, to be honest. That's how our universities operate and agree to keep us around. But those amount to, somewhere between $2 and $3 million for an R01 grant. And so our $200,000 investment is often leveraged into these $2 to $3 million grants. And that buys scientists a little bit more time to really prove that their methodology works or to find the answer to a question that could then move forward.
Awesome. Thank you.
I thought I might jump in on this as well, because we're talking about NIH funding, but if we're talking about developing therapies, if there's pilot funding from from ASF, and that leads to kind of a discovery of something that could be developed into a therapeutic, it is incredibly expensive and requires a lot of specialized knowledge to move a treatment into a human clinical trial.
Yeah.
I mean, you're talking about like easily $5 or $10 million or more to move something into a clinical trial, and that's where industry typically comes in. Pharmaceutical industry, biotech industry, and they're not going to just invest in an idea, but if you get some pilot funding, from something like ASF, to get the work going and to generate some initial data that this has promise, then that can attract the investment of industry to come in, and that's really where things accelerate towards actually bringing a drug to patients.
Awesome. So Mark, I'm going to direct this at you. So take us back to like the very, the beginning. You're, as an academic researcher, how do you take an idea or a discovery that you've made in the lab that you think might have potential, and how do you take that to the next step? Like what's that initial next step?
Yeah, I mean, ideas are relatively easy to come by. Ben and I, for example, we have lunches almost every day, and we come up with crazy ideas left and right. But I mean, ultimately, you have to look at the constellation of cool ideas and just the resources that are available. How much money do you have to do X, Y, or Z? And then even if you want to do one particular project, it's ultimately, I guess the dirty little secret is neither Ben nor I actually work in the lab. We have people in our labs, postdocs and graduate students. And so they're already doing a lot of things in the lab. And so we have to not only have a great idea, we then have to convince ourselves it's good, but then convince someone in our lab that it's worth doing in addition to the other things that they're doing. And so that's typically how things get started. And they usually get started pretty small as a side project because projects often don't work, and people know that in science. And I think you see this time and time again. And when it comes to the scientific discovery, like 9 out of the 10 times you try something, it doesn't work. And so you have to really be persistent, try things a different way, and then eventually something works. And it's been my experience that once something works, it usually flies, and things just sort of like come into place. It's like the topoisomerase discovery. It took forever to find that initial drug, but once they found one, we found another and then another, and the project has sort of sailed through. And so I think that's typically the way things work in science.
So this isn't necessarily obviously specific to this topoisomerase class of medications, but once you have something that you feel like is ready for commercialization or might attract the interest of a commercial partner, what does that process look like?
Well, that's also a lot of luck. So like the CRISPR therapeutic that we're developing, we talked to many different investors and drug companies and gene therapy companies, and every group or investor has a different risk tolerance. And I think it's pretty, in general terms, most of what comes out of an academic lab is pretty early stage. And so you have to find a partner that's willing to put in, as Steve Gray said, millions of dollars to de-risk it to the point where you could move it into a clinical trial. So it takes a lot of time from the pilot funding and an idea to an NIH grant to sort of build the preliminary data to ultimately in convincing an investor or a company that the discovery is worth moving forward into a human.
Great. Maybe for Ben, once you've convinced an investor, you have a commercial partner, what are those, I mean, we're still not at the finish line. You're still not in first in human trials. So what are those additional steps that have to be accomplished?
Yeah, I think what's really great about getting to the stage where you can partner with the pharmaceutical companies, I think the irony is it's something that I probably used to shun a little bit until I realized kind of the advantages that pharma provides and biotech provide, but they have a lot of expertise. I mean, they have taken things across the finish line, and most academic researchers haven't. So they know what's involved as far as developing a strong safety profile for whatever you're taking forward, for what you need to do for pivotal studies to de-risk it, what's pivotal for understanding efficacy, and also knowing and understanding the data package you need to be able to bring forth to the FDA to show them that you have something that's both safe and efficacious for use in humans at the target age you want to use. So there really is a lot that needs to be done. And working with pharma and biotech really enables that. They have a lot of expertise that as academic researchers, we shouldn't be shy about relying on and making kind of intelligent partnerships with the right company to move something forward, because there really is a lot to be gained and a lot of expertise that we can draw upon from that.
Yeah. Steve, what do you think the role of the patient community is in this therapeutic development process?
I think all of us could answer that, but
yeah,
I would just throw out there for one of the things. I mean, we need to understand what the patients most need, what kind of disease modifying aspects are most important for the patient population and their families. So I think interacting with the patients and their families is incredible. They're a great resource. And, one of the things that any family can do, even if they don't have money to donate to research, they can donate knowledge about what their needs are and be part of patient registries. Those are incredibly valuable to do. And Angelman Syndrome Foundation has a great patient registry. These types of things are really, really important, and they help get the pharmaceutical companies engaged. So I know in my lab, one of the things I do with every single person in my lab working on Angelman syndrome is they spend time over at the Angelman syndrome clinic, because it's important that they, at the very least, at least once, they need to spend a day with a family with Angelman syndrome, and to see the individuals and to see how wonderful these individuals are and how wonderful the families are, but also the challenges that they face. And I think this is really important. I think that's also why the family gatherings that the Angelman Syndrome Foundation has are so incredible, because we get a chance to interact with all the individuals with Angelman syndrome and their families. And not only is it inspiring to us and motivating for us, making us want to kind of move faster and better and safer, but we also understand what is needed by the community. And so those family conferences are incredibly important.
I chuckle because members of your lab have come to my house on a Saturday and spent brought lovely bagels, but spent the whole mornings with Evelyn and our family to really get an immersive experience of what that might be like. Anybody else that cares to chime in on kind of the role that the patient community can play in moving things along?
I guess my lab involves them somewhat more directly, although not always in such huge numbers, right? We've asked for samples. And what's funny is that I've not had to ask very hard, and I have people volunteering to give samples. And so that's been hugely helpful. And some of these samples have been really serendipitous in telling us important things and being useful tools in our lab. But then I think the other really important thing that, and I think Ben said this, that their participation in registries, but like the Ladder database, where this is a database that Liz really helped put together and helps curate and really has given it legs. It's so important to have this data, this clinical data, so that we understand their trajectory of how individuals with Angelman syndrome develop. And this will be really important for clinical trials. And so patients, families being willing to be involved, I know it, like they have to truck their kids and disrupt their lives to go and do these things. And that's, I'm in awe of that, because I don't know if I could, right? And so I'm always in awe of the families for doing that. And I think that's a really, really important undersold role for families in helping research.
Yeah, we have an incredibly activated community for sure, an educated and activated, very proactive, motivated community who wants to contribute.
And we should probably give a shout out to the registry, because I think the website is www.angelmanregistry.info. So www.angelmanregistry.info.
Awesome. Perfect. All right. With about 10 minutes left here, I'm going to lob out the $5 million question, as I'll call it. All right. I'm going to start with Eric. If you had $5 million to spend on Angelman research today, if I was just gifting you $5 million, what would you do with that money?
Great question. I'll use it for research. I guess that's the first.
Great. I was going to say it has to be on research, not like a new Porsche.
I'm very excited about some of the insights that we can get from these human stem cell models that I'm working on with Stormy, and I think that one thing that a good amount of money could help do is to parallel track the work that we want to do in the human stem cells with the mouse models, because I think there's a lot of opportunities for interacting there that are harder to exploit when people can only get large enough grants just to do their individual thing. But I think that some insights that we might find into signaling pathways or molecules and even other genes that we can identify in these patient-specific neurons. So we have great ability to learn some really detailed things there. But then that needs to really be validated in mouse models and other animal models, because in those animal models we have a better ability to look at higher order circuits and of course to look at behavior and make sure that we're looking at meaningful endpoints in terms of potential therapeutic opportunities. So if we could use the money to really do those efforts in parallel, identify what might be the best next generation of therapeutic targets to look at, and then develop cell-based assays based on maybe some of these individual patient lines that we could really use to screen, and not only screen in general, but also screen in a patient-specific manner what types of manipulations might be best. So just let me know when the money's coming and I'll start getting that in set up.
Fabulous. Your check is signed. Great. All right, Steve, same question to you. If you had $5 million today, what would you do with it for Angelman research?
Oh boy. Well, you know, it's tough because, you know, we've got some gene therapy approaches that I think we have, you know, we're excited about, that we're developing, you know, but it's still, you know, a number of steps that need to be done to look at, you know, is it something that could be applied to humans? Is it something, you know, that's ready? Do we understand all the risks involved? You know, there's still a lot of development work, but, you know, when we talk about a number like $5 million, I mean, it sounds like such a big number, but, you know, I've had things from my lab for other, you know, other diseases that we've worked on that we've actually moved into clinical trials. And $5 million does not get you as far as you think it will. And when you talk about, you know, something like a gene therapy approach and moving it into, you know, a real clinical trial, when I think about $5 million, I'm like, maybe if I use that to attract a matching donor, then maybe we'd have enough, you know, but
I think that's a really important perspective to put into perspective, that 5 million isn't enough to move the needle necessarily to get that across the finish line. I think, you know, these numbers are a little mind-boggling if you're not looking at these numbers all day, every day, and really appreciating the cost of moving these therapeutics.
Yeah, and you know, I think that that's something you know, that I kind of mentioned before, and you know, Ben said that, you know, he's working with AskBio, you know, to look at the possibility of moving a gene therapy approach forward, and you know, that's where you do attract those kinds of dollars to help move something along. You know.
Yeah. Stormy, if I gave you 20 million, what would you do with that? I'm kidding. I'm kidding. You have to work with the same 5 million as everybody else.
Damn it.
Sorry.
Well, I would look at my fine colleagues here and say, no,
I pool all of your 5 million into one pot.
Exactly.
Together. There you go.
And we would move mountains.
Okay.
No, you know, something that I've wanted to do, and it sort of builds on the lines of some of the stuff that Eric has talked about. I'd love to put together a phenotypic battery for our human stem cell derived neurons. Because first of all, we could run different therapeutic approaches through them to see how well we can evaluate therapeutic approaches using the stem cells, with the goal that if you could do it well in human cell culture, maybe you could more rapidly get drugs to the clinic. And then secondly, you know, I think about some of the stuff that Jason Yee does, right? We could plug in various patient variants into our cell model and ask, do they really cause Angelman syndrome? And if so, where on the spectrum does this lie? I had another thought about that, but I probably forgot. Screening technologies, right? So, you know, through some of our work, we figured out ways to make cell models that might be helpful for drug screening. And so maybe we could take those and also use them to look in a more unbiased approach along the lines with what Ben does. And like we could use it to support, I think, some of Ben's work more easily. And so I think that's what I would do with $5 million. I'd share and I'd partner with these guys because they're all brilliant.
Well, as a geneticist who's looking at all those variants of uncertain significance, I'd love a functional assay. So that would be really lovely if you could do that. Yeah. All right. Ben, how would you spend it?
Yeah, I think it's a really hard question. I think by and large, as a community, the community is doing the right research and asking the right questions for the most part. And I think it really becomes a matter of accelerating a lot of programs that are existing to try to just be more aggressive with them. So trying to get to those pivotal studies for safety and efficacy, trying to understand more about mechanism of action. And these are mechanisms by which drugs are unsilencing UBE3A, for example. And you know, this holistic approach by moving forward as aggressively as you can while still maintaining, you know, the safety standards and the standards to make sure you're bringing forth the best product, because that's incredibly important. Moving forward, the projects we have with gene therapy and small molecules and trying to get to pivotal safety studies, especially ones in non-human primates, I think is where I would really try to push that money.
Jason.
So with 5 million, honestly, I think it would be on the side of methods development. So I think, in the past several decades, our capacity for sequencing, so nucleotides, has really advanced. But I think our methods for analyzing proteins on sort of a large scale really haven't kept pace with our ability to sequence. So I think I would really focus on trying to do that, because ultimately UBE3A is an enzyme that degrades proteins. And I would try to develop some of those methods so that we can maybe come up with things like biomarkers that may be important for Angelman syndrome and that may be used for some of these therapeutic development studies and for diagnostics. I think that's what I would do.
Awesome. I'm ready to write a lot of checks tonight. Just kidding. Mark, last but not least, as always, sorry.
So I guess I'm last again.
Again, sorry.
All right. Well, yeah. So I guess I'm pretty optimistic about where the therapeutics are with ASOs. You've got 3 different commercial entities that are moving these forward. So I think we talked about how discoveries and how ideas get turned into drugs, and so this is an example of an idea that went from seed funding to NIH funding to drug companies, and now there are 3. So millions and millions of dollars are being spent on ASOs, and so we're going to know in the not too distant future if that's going to work, at what age it's going to work, and so I'm kind of thinking, like, in the future with $5 million, how could we really move the needle in the field? And I really think what we need to think about is like where many of you were 5 years ago, 3 years ago. When you're thinking of having kids, you never thought, probably in your wildest dreams, that you would have a child with Angelman syndrome. And yet we know what the gene is. It's due to loss of maternal UBE3A. There are ways of diagnosing that prenatally. And so I really do think the next step in the field is to try and detect it as early as possible. Maybe we've had kids. We did some genetic testing. It's becoming more and more common these days. And I'd say in another 3 to 5 years, half or more of all people who have pregnancy are going to be tested genetically. And the technology is getting to the point where you can actually pick up the deletion that causes Angelman syndrome. And so I want to be there in 5 to 10 years with a therapy for these families who are, they don't yet have a kid with Angelman syndrome, but they have a baby, an unborn baby with the mutation that will cause Angelman syndrome. And I want to have a therapy that's going to work for them so that when their child is born, the child does not even have Angelman syndrome. And so with the $5 million, I would advance that idea in that direction.
I like it, outside of the box. So prenatal screening, newborn screening. I think these are critical areas, and I like that we're, thinking about those as well. So this has been phenomenal. I really appreciate all of you for taking the time and letting us pick your brains and learn a little bit more about what's going on inside your labs and the exciting work that you're doing. I would certainly be remiss if I didn't also give a huge shout out to the other heroes here, which are our families who are grassroots funding, the money that's critically needed, as well as FAST and our other private foundations who are funding really important work to make these treatments a reality. So we appreciate that this is not something that we can accomplish alone. We love this team mentality here. I love how much all of you on this call are working together towards the same efforts. And so we're excited to see where the next 5 to 10 years takes us, and looking forward to hopefully hearing more from each of you all at the Scientific Symposium in August. So thank you all. Have a great night.
Thanks.
Great.
Thank you.
Thank
Thank
you.
you, everyone. Thank